Hey everyone, thank you for coming along. Mark Stafford from ZEISS is the Product and Application Specialist and is very generously giving us a talk today on the new system that we've got, which is calling it ZEISS Widefield Live Imager 2, because we do have a number 1 system that we're developing as well. It's not quite ready yet. And we have the Fluoro 2 system, which will be somewhat relevant to some of the parts of this. And Mark's going to talk about live imaging a bit more.
Yeah.
That's also not Fluoro 2, but some of the stuff in Zen is applicable. Not the same version of Zen.
Some is. Yeah.
I'll be also on the chat for the Zoom people. And if you have questions, do you want them held at the end? Or
I actually don't mind people interrupting me, actually, to be honest, but it's a fairly long presentation. Yeah. So let's just see how it rolls, to be honest. Like, I'm happy for people to interrupt, but yeah, whatever. So thanks everyone for coming. I am just going to be talking about live cell imaging in general. So sort of key considerations for best results I put up there, but I also wanted to introduce into a little bit more depth the Observer 7 live cell imaging system that you have up on level 6. That's an exciting system that was installed, I think, in December last year. So I guess it's covering some of the key features of that system so that you have an idea of what it can do for you. So just to give you a bit of an agenda of the talk, just going to be talking about how we keep our cells healthy when we're imaging, minimizing sample damage, choosing the right sort of objective for the task, how we're able to maintain focus during live cell imaging experiments, camera types for microscopy, what are applicable cameras and the technology of the cameras in specific relevance to live cell imaging, and then looking at the Observer 7 imaging platform in a bit more detail, and just talking a little bit about optimizing experiments and just generally what the system can do for you, as well as covering some of the typical software modules, but some of the more esoteric software modules that are available to you as well. So where did it all begin? From the point of view of ZEISS, the sort of the birth of live cell imaging, if you like. This is actually what's called the microcinematograph, which is an awesome name for a microscope. It was invented in 1955, and it was used to image the first phase contrast movies of living dividing cells. And you can see some familiar sort of aspects of, I'm trying to get the flow of this thing to work. You can see some sort of, I guess, sort of familiar aspects to this microscope. You can sort of see the microscope here, obviously, the old ZEISS standard, but around it, you can see this Perspex box, which is the incubation box for the microscope. You can see the controls, the focus controls, which come outside the Perspex box for focusing. And this device on the back here, this very large device, is the camera for recording the images. Of course, camera technology has changed a bit since then, which is pretty exciting. So from 1955, we come all the way to 2021. And this is your system on level 6 at the IMB. As I said, was installed in December last year. And basically, I would say this is one of the very high-end live cell imaging systems that I've installed of recent times. It has a lot of bells and whistles on it to enable you to do some pretty exciting stuff for the sort of work you're doing here at the IMB. So at ZEISS, we cover pretty much the entire, we have an extensive and very complete microscopy portfolio where we could sort of cover everything from the macro or larger micron scale all the way down to that sub nanometer scale with our helium ion microscopes. And today we're going to be talking with where the live cell imager fits in is in wide field light microscopy. So live cell imaging, basically it's obviously observing dynamic processes within living cells or whole organisms. And I just want to talk about what do you need to consider when examining living cells under a microscope. Because when you're doing live cell imaging, you're essentially looking at dynamic processes Okay, so that's the key word there, dynamic processes that are cellular or subcellular in structure. And you need, and these are the key points really, you need good temporal resolution, so good time resolution, and of course, good spatial resolution. And the other key point here is we also need to maintain, of course, a physiologically relevant microenvironment so that we can basically determine or deduce correct information from our imaging experiments. So you want to keep our cells happy, essentially. Otherwise, your results are going to be certainly skewed. So some key points to consider. Living cells are sensitive to damage, obviously, so we need to be careful of that. Image signal is precious. Okay, photons are pretty hard to find sometimes, particularly in live cell imaging. So we need to have good signal to noise when we're doing these sort of things. Often the best cells or the most important cells are the most challenging to image experimentally. So what can we do to give ourselves the best chance for success? So key points: sample health, spatial resolution, temporal resolution or time resolution, and signal to noise are key things that join all together when you're doing live cell imaging experiments. They're the main things that we need to think about, and they all kind of interrelate with each other, obviously. So if we look at keeping cells healthy, you know, some cell death is normal, but we've really got to watch for excessive signs of cell death or cell stress when we're doing our live cell imaging experiments. And I guess comparing cells that are not imaged, so cells that are sitting in an incubator, is a good reference in this case. So we can look at how the cells are going in the incubator versus on the microscope and see whether they are correspondingly healthy. So you're really, for the best experiments, like the cells should pretty much grow on the microscope, or the ideal situation is that the cells will grow on the microscope just like they do in the incubator. We need to recreate that environment as best we can. So the first key point is heating. So you would have seen on these microscopes before, potentially, that we can, of course, most cells require around 37 degrees or 37 degrees for optimal cell growth. Some may be slightly lower than that sometimes, but generally 37 degrees. So we can have like a heated, a specific stage insert that goes into the microscope and a cover that ensures the sample temperature is maintained and that it creates like a nice humid microenvironment on the microscope itself for the cells to grow in over an extended period of time, hopefully like they would be if they were in an incubator. Okay, so this is a critical aspect of live cell imaging, to keep your cells nice and happy on the microscope while they're being imaged. Another thing to consider is pH. So of course, when you put your cells in an incubator with the culture media, that culture media has particular buffers in the media itself, and those incubators have CO2 flowing into them to maintain or buffer the cell culture media chemistry. Okay, so we achieve that by these special gas mixers, which are controlled by the microscope and the software to pump a particular level of CO2 into that small microenvironment. If required, and this doesn't necessarily pertain to your system, but certainly other systems, we also have the ability to control oxygen concentrations. So some people want to look at hypoxic conditions when they're doing live cell imaging, and we can manage that and control that by displacing oxygen with nitrogen gas on the system as well. This can be combined certainly within the one experiment if you wish to do so. Humidity is a big one. We don't want to have our cells evaporating or the culture media evaporating when we're doing long-term time-lapse experiments. So can we change that thing on the top, or is that kind of, okay. Yeah, so to keep a humid environment, we have a special humidity device that sits outside the microscope. It's basically just a warmer, essentially just a heater, and we pump that CO2 gas through that heater device. Sometimes you see just the incubator bottle in the microscope incubator itself, but there's no control over that. It just sits at the one temperature. Okay, it's much better to have a specific humidity device outside the microscope where we can control the temperature of that. So essentially, the higher the temperature up to a certain point, we can reach a sort of saturation level of humidity and optimize that quite precisely to prevent evaporation of your sample. Just looking at, again, looking at minimizing sample damage. You all know that the electromagnetic spectrum here, and you would all, as microscopists yourselves, you would already know all of this already, but just to reiterate, shorter wavelengths of light have more energy, longer wavelengths of light have less energy. We're generally looking at the visible spectrum here. When we're imaging, so between 400 and 750 nanometers. General rule for live cell imaging or imaging in general really is that the longer wavelengths of light are less damaging to biological organisms and cells than shorter wavelengths of light, just like we're more sensitive to UV radiation than longer wavelengths of light, okay? On another note though, just for some light sources do give out infrared radiation. Longer wavelengths of light, such as like infrared, can also cause sample heating. So avoiding shorter wavelengths of light and infrared radiation are beneficial for imaging living cells. And if we look at the light sources that are available, generally for microscopy, I mean, I guess you probably don't see too many cases at the moment of mercury arc lamps or xenon arc lamps. You might see microscopes currently with metal halide light sources, but most commonly you'll see these days microscopes that have specific LED illuminations for fluorescence microscopy. And LED illumination is certainly the most gentle type of illumination for live cell imaging applications. So when you look at how light damages cells, essentially it's just has the ability to cause the creation of free oxygen radicals, and free oxygen radicals can be very damaging to living cells. Okay, so light is a direct catalyst for that issue that we have when we're doing live cell imaging. There are a few ways that can be mitigated. I've never really tried this. I think you've probably tried it. Yeah, so I know it's unusual to see people doing this, but I think it's worth mentioning anyway. Particularly if you have very difficult cells to image, it's always a consideration that you can think about. So I might as well put it up here and just talk about it a little bit. So you can use oxygen scavengers to soak up those free radicals. I would only do this in extreme cases pretty much. But things like propylgalate, oxyfluor and Trolox are chemicals that can inhibit free radical formation and maybe keep your cells a little bit healthier. But sometimes these chemicals can also perturb normal cellular processes. So it's a bit of a balance whether you use these or not. Some molecules in cell culture media themselves have been shown to reduce photostability. For example, in GFP, you can get media, say DEMEM media, that lack certain molecules like that and are more suitable for live cell imaging. So consider the media that you're growing your cells in is an important consideration as well. So things like riboflavin and pyridoxal can decrease GFP photostability. However, other molecules like rutin, I've never heard of that, can increase GFP photostability. So think about what's in your media as well as the bottom line of that when you're doing live cell imaging experiments, and pick media that is suitable for that application. If you can, and this is not always possible, but choose bright photostable fluorophores whenever possible. Haha, it's not always possible, really, to be honest, in the real world. But if you can, choose fluorophores that are more photostable, do so. Choose fluorophores that have that you image in preferably longer wavelength illumination. Obviously, that's going to be less damaging than imaging in the shorter wavelengths of light. If you have to use fluorophores that need that UV or blue excitation, You can think about how you set up your experiment. So things like increasing the interval time, reducing the light intensity, and keeping your experiments short. So the key thing about live cell imaging is essentially to use illumination with caution, and not all imaging modes, the way that you set the microscope up, strictly control that illumination versus image capture. Okay, it's kind of an important consideration here. LED illumination is really good because you can adjust the light intensity very precisely per channel, okay, to reduce that or minimize that potential for sample damage. The other thing that you can do, particularly for these high-speed systems, these high-speed live cell imaging systems, such as the one that you have, is to use what's called trigger control. So this is just an example of a trigger control box, which is able to synchronize the operation of the cameras, the filters and shutters, and the light sources all together. So you sort of, I guess, synchronizing the movements of the filters, the light sources, and the camera all in at once, and that can make the system itself operate at a very high speed. Okay, so it's also good for high temporal resolution. Okay, but it's also good for minimizing any excessive light exposure to your sample, because you're essentially triggering all of these components exactly when the camera is acquiring. Okay, it's all synchronized to the camera. As soon as the camera is doing something, all the light sources are doing things at the same time. Okay, and it can make the system operate very, very quickly. Okay, which is also an advantage. Choosing objectives. So of course, choosing the right objective is a very important consideration for any kind of microscopy application. You know, you have to choose the appropriate objective to get the right sort of answer. But for live cell imaging, I guess some caveats there is you want to choose an objective that has a high light gathering power. So generally using an objective that has the highest possible numerical aperture with the lowest possible magnification, if possible. Okay, so if we look at the 40 times objective here, and we have the same NA for the 40 with 63 and 100, you can see the light gathering power decreases as we go and increase the magnification. Okay, this has got the same resolution, but the light gathering power decreases as you go up in magnification. So again, it depends on what you want to do, but if you have a high light gathering power, you can have shorter exposure times, the system runs faster, and your samples are exposed to less light, essentially, or you can use less light, which again assists with minimizing sample damage. Minimizing spherical aberration is also an important consideration, both for fixed tissue and live cell. So you can get a refractive index mismatch between your immersion medium and your embedding medium. This can reduce contrast, resolution, and brightness. So by getting those things as close as possible, you can again increase your light efficiency and get better imaging quality. Just again on the spherical aberration side, so objectives are generally designed for use with 1.5 type cover slips. These are 0.17 corrected cover slips. If you use cover slips that are the wrong thickness, you're again going to compromise your imaging optimization, again reducing brightness, contrast, and resolution. So think about the cover slips that you're using, use good quality cover slips, and of course some objectives you can have correction collars on them. You would have seen that. So if you are using objectives that do have correction collars, use the correction collars accordingly to minimize your potential for spherical aberration. If you're using long working distance objectives on the system for your cheaper plastic plates, now I never really advocate that for high-end live cell. You want to use the cover slip thick bottom plates, the more expensive ones. Not everyone has that choice. Sometimes you do want to put a cheap plate on there, in which case you're probably going to want to use one of these long working distance objectives. So you'll need to change that correction collar accordingly to the thickness of the plate that you're using to minimize that spherical aberration. Okay, if you want to go deep into the sample, that's another consideration. So resolution falls off quite dramatically with depth. If you're doing live cell imaging experiments, or say looking at zebrafish or something like that, perhaps think about the objectives that you're using. You may want to pick something like a water objective, which will have a better ability to go deeper, okay, and give you better better results. Now, I guess you guys probably do have some water immersion objectives available. I mean, the system's configured with some objectives, but it doesn't mean you can change it if you need to. Maintaining focus, of course, is difficult over long-term time-lapse experiments. It's something also to consider. On a simple sort of side, you can, if you look at high magnification objectives, they have a very narrow depth of field, so you'll see focus drift quite quickly and quite easily. You'll notice that very easily, as I said. A simple solution is you can choose a magnification, lower magnification, and would decrease that visible drift. Or, of course, for a high-end system, there's another solution where we put a heated incubator around the microscope itself. And a lot of people think the incubator is actually for keeping your cells healthy and happy. It's actually just to keep your microscope at a desired stable temperature, which minimizes thermal drift between your objective and your sample. Another thing that can be added or tried out is we do have special live cell imaging objectives, these LCI objectives that have a special insulation ring installed at the top of the objective there, which minimize heat loss and also minimize thermal drift between the objective and the sample as well. Hardware focusing could also be used. So, for example, using something called, in our case, definite focus. I think Nikon have something called perfect focus. So each microscopy supplier has a different name for them, but essentially it's a similar sort of principle. It's basically a device that detects the bottom surface of your sample, it's actually looking for a refractive index change between the glass and your sample. If it can't find that refractive index change, it doesn't work very well. So it basically generates a reflection. You can set a particular point in the software to say, always stay at this particular focal plane. It's basically just bouncing light off the surface of the dish. So, okay, you've got a detector here, you've got some optoelectronics here and your lens, and it's basically finding that a particular focal plane by reflecting off the surface of the sample and cover slip there. It's very fast, actually. It's a very good way of doing it for live cell imaging. For tricky situations, you can also combine this hardware autofocus technique with software autofocus. Sometimes you might have samples that aren't always sitting in exactly the same plane on the bottom of the cover glass. They might actually move around even a little bit. And you can combine hardware autofocus to go, okay, go to this point, and then do a short autofocus above that to find my sample that might be moving slightly out of that focal plane, okay? So remember that you can combine these focusing strategies together in the ZEISS software. Cameras. Okay, so camera technology changes all the time. There's many different types of cameras around. You would have heard cameras with different chip types. So for example, CCD cameras, EM CCD cameras like you have on the spinning disc system. So they're very, very sensitive cameras, and CMOS cameras, and scientific CMOS cameras, which are, I guess, going to probably be the dominant cameras moving forward for a period of time. Basically, the CMOS chips have replaced the older CCD camera technology. They're no longer making CCD chips anymore. Everything's sort of moving to CMOS chips now. These cameras are extremely fast. They have low noise. They did suffer from high noise for quite a period of time, but with new technologies coming out, they've really made some huge inroads there. And the cameras coming out now do have very low noise, low noise capabilities. They have a high pixel number, and they're very, very well suited to microscopy applications. Just to talk about quantum efficiency. So I'm just going to talk about some camera parameters generally here that you might hear how they're describing a particular camera, I guess. So quantum efficiency is basically looking at a measurement of the device's sensitivity to light. It's usually expressed in the spectral range, or sort of around the green to yellow level. So if you look at standard CCD cameras, they're around 70% quantum efficiency. EMCCD cameras are really high, around 95, and SCMOS cameras that are coming out now are also really up there, so around 80 to 95% quantum efficiency. It just means they're very sensitive and can detect low levels of photons. Okay. Some definitions about cameras generally. If you look at a pixel here, light will strike the semiconductor material on the pixel. This generates charge, or charge builds up inside the pixel, which is then read out by the camera's electronics, so a readout amplifier, an analog digital converter to your computer. The full well capacity of a camera is basically the total number of pixels that that can be collected in this bucket, if you like, in this pixel bucket. Dark noise is something that occurs when the camera warms up, and that's why you find a lot of these cameras will have active cooling down to -20 or even -80 degrees by Peltier cooling to reduce that dark noise. Readout noise is basically just, I guess, the noise of the electronics themselves. And the dynamic range of the camera is a function of the full well capacity and the readout noise and dark noise. Okay, so the idea is to have a camera with a very low noise readout and a low dark noise with a good deal of capacity to collect a lot of electrons, which essentially gives you the dynamic range of the camera. So it's those sort of parameters that people try and optimize when they design a particular camera for scientific work. Bit depth, you'll see also cameras that have different bit depth ranges. I guess if you have more bits, so you have something like a camera that's, say, a 12 or 16-bit camera, you're going to have a better and more accurate representation of the intensity values of your sample. Okay, so generally speaking, like color cameras, if you're doing color imaging, 8-bit camera might be okay. When you're doing fluorescence imaging, having a camera that's around 16-bit would be an advantage for you. So megapixels is not necessarily always better. So more megapixels in a camera is not always a better thing. It's great in your phone. Sorry, I got a dry mouth. It's great in your phone, but it's not always applicable in microscopy. So what we really look at here is if we look at pixel sizes, for example. So small pixel camera with small pixel sizes is good for high resolution imaging. It has a lower dynamic range and it's less sensitive to light. Okay, if you have a camera with larger pixels, like an EMCCD camera, very, very large pixel size, very sensitive to light, it can be less noisy. They're not so great on spatial resolution, but very, very sensitive. So I guess what I'm saying is that pixel size of the camera and the size of the chip, it's a balance between sensitivity, larger pixels, and resolution, smaller pixels. It's a compromise to give you basically the best suitability for a given optical setup that you're using. So the choice of camera and the pixel sizes and the sensitivity, all of those things come together for a particular application. So for live cell imaging, you want to have pixel size that's not too small, maybe not too large, and just about the right sort of size to give you a good compromise between temporal resolution, so speed resolution and sensitivity, and spatial resolution, sorry, rather. And that's just sort of outlined here. So if we look at Mr. Abe, okay, the famous ZEISS guy, he will tell you the resolution that's present in the optical system of the microscope, okay, how we can differentiate two objects from one another. And Mr. Nyquist here, he tells you how many pixels you need to avoid loss of resolution when you acquire this image digitally, okay? That's gonna be slightly hard to explain, but you need to basically sample that by at least 2 pixels to satisfy that Nyquist criterion, okay? If you undersample that, you might not be collecting all of the information from the microscope system, from the optical system. Okay, so the rule here is basically the larger the magnification, the smaller the required camera resolution, okay? The larger the NA of a given objective lens or a given magnification, the larger the required camera resolution, okay? So to satisfy this Nyquist criterion for being able to capture the information that the optical system is giving you, that the microscope is giving you, it's a balance between your camera's resolution and your objective choice and parameters. And it really depends on what you want to do. So Nyquist sampling is important for something like, for example, deconvolution, okay, where you need to collect the right amount of information from your optical system to be able to process that image mathematically, all right? It's not always possible in live cell imaging. Sometimes it's a bit of a compromise, okay? So think about your camera parameters and your camera and the objective you're using and what you want to get out of it, okay? So if you want to do something like deconvolution, you have to think about your camera resolution. And sometimes it doesn't really matter. It really depends on what you want to do. Well, it doesn't matter as much, and you still need to think about it. So for example, if you have a camera like the prime BSI camera from Photometrics, and this is actually the camera that you have on your system. If you're using the Planapo 63 1.4 oil objective, which you also have on your system with no binning, this is actually perfectly sampled according to the camera's pixel size, 6.5 micron pixel size. If you're using a 10 times objective, 0.45 NA, same camera parameters, this is actually not capturing the full resolution of the microscope, okay? It's not the end of the world, but if you're using something like, or you want to use a technique like deconvolution, then that might be a little bit compromised for Nyquist sampling. The alternative would be to use an objective that has a slightly lower NA. Okay, there's too much information for this particular talk, but I just want to get it in your head to think about the objective choice and your camera parameters a little bit if you want to use some of the processing tools available to you at the IMB. And I've done this myself. I've used the wrong objective, the wrong camera parameters, and then I've tried to do deconvolution and it's looked not that great. And then you try it with a different lens, different camera parameters, and it looks fantastic. Okay, so it's not magic. You've just got to think about it a little bit because the camera's a fixed thing. The pixel size is fixed, you know, just think about how you want to apply that. Sometimes it's just more sensitivity, but sometimes if you want to do that extra processing, you have to think about how you're acquiring your image in a bit more detail.
Let me just say, like, if you're going to spend time optimizing the staining, your labeling, your imaging, don't skimp on the acquisition either. So think about the experiment. Talk to us. You know, I talk about this stuff. It's our, you know, bread and butter. So there's nothing worse than someone coming to you with all this data and you go, just spoke to this beforehand. It'll help you put down. Especially from the group leaders point of view, you don't want to be, I think, people using microscope time, paying for it, and not being able to get the.
Yeah, especially if you've done a week-long experiment, you've generated a ton of data, and then you can't deconvolve it later because you used the wrong objective or the wrong camera parameters. I just want to touch on binning as well. So binning is a technique where you can combine pixels together to make the camera more sensitive. Okay, so got one pixel here, 2x2 binning, you combine those pixels together, get one larger pixel, 4x4 binning, and indeed a larger pixel again. So this can make the camera really, really sensitive, okay, and faster as well. But use those parameters cautiously, because if you do bin, there's a price to be paid as well for binning, okay? So you can see that one good thing about it for live cell imaging is it can really reduce your exposure times and cell phototoxicity and photobleaching. But it will reduce your image size and resolution as well. There's always a compromise. Okay, so just think sometimes that's fine as well. It really depends on what you're trying to see. Okay, but I think some people they go to the microscope and just the cameras are always the same parameters. Okay, just bear in mind that you can change those things around because it's always a compromise between resolution, speed, signal to noise, and sensitivity. It's always the triangle of compromise when you're doing these experiments. So I just wanted to get on to the system that you have here up on level 6. So it's a fully motorized Axio Observer 7 microscope. So this is the latest inverted imaging platform we have at ZEISS for Widefield and fluorescence and confocal imaging. You have a few of these around already. It's got full incubation, CO2, humidity, and temperature control, a Colibri 7 LED light source, 7-line light source for fluorescence applications, an LED light source for transmitted light applications, motorized XY stage, high-speed filter wheel, high-speed Z piezo focusing device, which I'll tell you about in a minute in a little bit more detail. It allows you to do very, very fast Z stacking. Multiple multi-band and single band filter combinations, which I'll outline in a bit more detail in a minute. Hardware focusing, so it's got that definite focus 2 hardware focusing device. It's fast triggered for high-speed imaging. It's got an apotome as well, apotome 2 for optical sectioning. It's running the Zen 3.2 blue software. And it has a photometrics prime BSI camera for fluorescence imaging, so very high-end SCMOS, scientific CMOS camera, and also a 705 CMOS color camera for bright field imaging. I just want to talk about the some of the system highlights, so some of the components on the system in a bit more detail. So let's look at the light sources. So the Colibri 7 light source that you have for fluorescence is again triggered for high-speed imaging, and the Colibri lends itself very well to that. You have a 7-line Colibri in your system here. So in the basically LED, multiple LEDs in the Colibri unit. You can see sort of the spectral characteristics. So it's not wide. You have like a band of excitation light from the device. LED, as I said, is very gentle for live cell imaging. It gives you excellent contrast. You can adjust the intensities of each of these individual LEDs within your experiment. So you can have one at a high intensity and one at a low intensity if you need to. The Colibri itself calibrates every time you turn it on. So the power that's delivered to each LED over the course of its lifetime is measured and remains stable. So from one experiment to the other, the light intensity of one experiment should be the same as the next week that you do that experiment, okay? So it's 20% should still be the same light intensity because it measures that and calibrates before it starts. So this is configured for, if you want to just look at general fluorophores, you've got a 385 excitation line, a 430, a 475, a 555, 590, 630, and 735. So it does that. If we look at just general fluorophores, say DAPI, CFP, GFP, FITSI, Sci3, DS Red, Texas Red, MPlum, something like that, Sci5 and Sci7 excitation. And this is also triggered, so it can turn on and off very, very quickly for high-speed image acquisition. Next in the, I guess, the fluorescence light train, if we just stay on that for a moment, we have a high-speed filter wheel in the system. That's one of these devices here. That goes into the filter turret. And essentially this actually splits up the sort of the beam splitter and also the emission filters on the system. So it's, as I said, motorized. There's a 6-position wheel for beam splitters. So you can have 6 beam splitters in there and a maximum of 8 potential emission filters, okay? And that emission filter wheel is fast triggered, so it moves very, very quickly and has a switching time of less than 50 milliseconds, okay? You can actually increase or decrease the speed a little bit in the software. So we've included some multi-band pass filters in your system for high-speed imaging, and also some single bandpass filters for more specific applications, I guess, depending on what you're trying to do. So in the system, we have a pentaband filter. So that's a 5-line filter, filter set 110. That's not really relevant, but basically a filter for DAPI, say FITSI, GFP, Sci3, Sci5, and Sci7, so 5 lines. And a quad band filter as well for, say, DAPI, FITSI, M-Cherry, and Sci7. So that 555 line and the 590 line are separated between 2 different filter sets, because you can't really have them in the same, or you wouldn't generally have them in the same experiment. And some single bandpass filters for DAPI, FITSI, Sci3, M-Cherry, and Sci5. Okay, so if we look at, this is actually written in your user guide for the system. I think it's written in your operating procedure manual. It's also in the software if you need to see what's where. But just to give you a visual representation in this wheel here, these little units here, position 1 is for DIC bright field. 2 is your pentaband filter, your quad band filter, and then we have position 4 to 8, where we have these band pass filters for DAPI, GFP, FITSI, Sci3, M-Cherry, and Sci5. Okay. So as I said, you can switch LED lines on and off really, really quickly in the experiment, which is great because you can go really, really fast. So if we combine that with, say, a multi-band filter, say the pentaband filter, something like filter set 110. So that just involves a beam splitter and 1 filter. So nothing's moving in the acquisition. You're basically just switching the LEDs on and off really, really quickly. So this makes for a really, really fast image acquisition. Okay, so you can get the system to go super fast because you're not moving anything. So that's great if you can do that. I guess it gives you maximum speed. I guess with multi-band filters and the more lines that are on the filter, there is more potential for signal crosstalk. So if you do get signal crosstalk, some bleed through, as they call it, you can, in this system, maybe think about using the single band emission filters to clean up your signal. Okay, so as I said, you've got 4 of these filters in the system. You can keep, say, for example, if we're using this 1 filter, so same filter set that I had before. We're not moving the beam splitter, but we can just move that fast emission filter wheel and capture there. I can't go back. No, go back. So you can just use the fast emission filter wheel to quickly change, keeping the beam splitter the same, and just move that emission filter wheel really quickly to capture your signal in a more specific way. Hopefully that makes sense.
The emission signal, the amount of light, is that comparable between the filters?
When you're using the single bandpass emissions, they're going to be a little bit tighter. They're going to be a little bit narrower. Okay. As my understanding, they're not quite as broad. It's hard to match them perfectly. We kind of do match them pretty well in that pentaband filter. I can't get it up at the moment. Yeah, that one, it's pretty well matched. So these are these emission bands here are really as much as we can get them anyway. The single emission bands that you guys got are they're slightly narrower. The guys did their best to try and match them as best we could. Yeah.
So say you have an experiment with 3 colors, low signal, but you don't care about time. What would you recommend?
I'd probably, if you don't get crosstalk, I would just use the multis. Yeah.
7:30 or something.
Yeah. Yeah. So think about that. So think about the separation. That's the critical thing.
Similar to, if anyone here is big on deconvolution train, where we call them faster. So if you want to go fast, you have the quad band, the penta, but you don't worry about that crosstalk.
I would
Localization as well. Like if you're worried about the DAPI stand coming through a little bit.
Or you can combine them. So you could do, say, oh, DAPI looks like crazy. I'll just go and put the single band in for that and then do the penta for the others. Or do the, you know, it's, again, it doesn't have to be one or the other. It can actually be a combination of them, depending on what your stains are. Yeah. Okay. So another aspect of the system is the LED light source for transmitted light. This is the VisLED here. Now, with this system, essentially, again, you want to go really, really fast, and we have the ability with no shutter inside the there is a shutter there, but we can keep it open. We put this special filter in here. This is a phosphorescence blocking filter that goes into the transmitted light path. When that filter is in place, we don't have to use a shutter here. Shutters are really slow, like they make it just a mechanical opening and closing. If we don't have to have a shutter and a component in there, we can switch really, really quickly between fluorescence imaging and bright field imaging, okay? Because we're just basically flicking on LEDs. Yeah, so we have that capability on the system as well. So high-speed imaging between fluorescence and bright field / DIC applications are available to you on the system. Motorized stage, you have obviously a motorized XY stage on the system, but you also have this Z piezo top plate. So the Observer 7 has a very fast internal Z drive anyway, but with this 500 micron Z piezo top plate that you have within the fact that the system's fully triggered, you can do incredibly fast multi-channel experiments. And with this device, this Z piezo device, really, really fast Z stacking as well, okay? So you can combine all of those modes together for very fast three-dimensional imaging reconstructions. Objectives on the system for fluorescence, DIC, and phase contrast are 5, 10, 20, 40 dry objectives, and you also have a PlanApo 40 oil objective and a PlanApo 63 1.4 oil objective as well. But you also have other objectives that are available at the facility up there. So if you want a water objective, I'm sure that one can be found for you to do maybe fish work or something like that, okay? The definite focus 2 module, I've already spoken about that before, so I'll just move on quite quickly. But again, this is a very fast way of doing focusing. I would recommend using high quality plates and dishes if you can. It will also work with plastic dishes as well, as long as it can find a refractive index change, but these do work very well on the system. So I'd recommend high quality cover glass if you can for best results. Okay, the question was in the software, you have particular controls on the right-hand side of the software. It can be a bit confusing as to which one you use. There's 3 controls on the right-hand side of the software. One is find focus or find surface, and that is quite neat actually. If you press that one button, it will just find the refractive index change and get you close to the focal plane. Not perfect, but just close to the cover glass. So then from there, you can just find your focus easy. There's one called store focus, which is kind of, I want this to be my focus point when I start my experiment. The one on the right to that is, yeah, okay. And then there's recall focus, which brings you back to a particular point that you just set. And there's lock focus, which is awesome. What is lock focus? So if you're looking around a plate like this one, right? So you're looking around a plate and you are on the first well and you lock the focus and you're just navigating. It's just for navigation. So it will just continually try and maintain that focus point that you set across the plate. So wherever you drive to with the stage, it'll maintain that focus point. Okay.
How is that different from store?
Because store focus just stores the focus. It's not doing it in real time. It does that for the experiment. Lock focus is when I'm just looking around. It's just purely a looking around thing.
And then in the I think it's called focus strategy.
Yes.
You also have to click which one you want to use, right?
Well, it will ask you at the start of the experiment to pick that first focus point that you want, right? Depends on how you set up that focus strategy, to be honest. Probably would have to show you on the instrument, but if you've already stored the focus, then you've stored the focus when you start the experiment. Essentially, there's multiple ways that you can do it. There's multiple parts in the software of doing the same thing, depending on where you are. Okay. Yeah, so we just got to move on quite quickly. Sorry, this is a pretty long presentation. So on the system, you have like the full incubator, as I mentioned before, what that's for. Obviously, for minimizing thermal drift, you have full independent control of the CO2 and humidity control here, which can be controlled on the microscope's touch screen or in the software itself. You have also the LabTech heated insert with CO2 cover on the system. This is a heated insert, so you'd want to use this insert for your long-term time-lapse experiments. So things that might be a week long or something like that, you're going to get a very, very stable microenvironment using this heated insert in combination with this particular CO2 cover. So this heated insert can do chamber slides, and 35 and 60 ml dishes. Yeah, you can run experiments for easily a week or longer if you want. So you can control all of those parameters in the software itself, and don't be afraid to experiment. Okay, so I've had certain cases of live cell imaging where you pick 37 degrees, but you know, it doesn't necessarily, you might tweak that a little bit to make it exactly right for your cells. Or CO2, 5%, maybe it could be 6%, I don't know. These things can be changed if you need to. Camera-wise, you have the BSI prime BSI camera. This has extremely low noise. It's actively cooled, so some of those parameters I was talking about before, this does really fit very well into those. The pixel size is 6.5 microns, which fits nicely for NIQUIS sampling for the 63 and 40 times objective lenses. It's best matched with the 63, but also the 40 is not too bad as well. The speed of the quantum efficiency is very high, around 95%, and the frame rates that you can achieve, it's around 40 to 63 frames a second in full frame. But you do have the ability to change the binning, frame crop, size, gain adjustment, things like that can also be changed. If you change the frame size, you can really change the speed that the camera will operate. And it also has some nice, I guess, denoising capabilities for enhanced signal to noise on the camera electronic processing as well. I need to check out how that would be applied in Zen. I haven't used those parameters, but it's definitely capable of that. So very good camera for live cell imaging. For bright field imaging, on the right-hand side of the camera ports, we have the 705 color camera. This is a 5 megapixel cooled CMOS camera. Very fast frame rate, great color interpolation. The pixel size is smaller. So the pixel size here is 3.45 micrometers. So again, smaller than the one for fluorescence imaging. But the reason it's an advantage here is that we want high resolution for this bright field work. Okay. We have the Apotome 2 also on the system. The Apotome 2 is a structured illumination device. It's a combination of optics and mathematics to generate an optical section. It projects a grid onto your focal plane, takes 3 to 5 images, and will generate an optical section from that. It'll work with all objectives. It's best with high NA objectives. I would recommend it only really for fixed tissue work. You can use it for live cell, but live cells that are not highly dynamic and moving around, because it's a bit slower on the acquisition side. You can take 2D and 3D stacks with the Apotome to generate an optical section. It gives you kind of like a confocal-like type of image. And you can use deconvolution within Zen to increase the resolution further if you wish. So this is just an example of how it works. And you would have probably seen this on Fluoro 2 as well, on the upright system. You have an apotome on that system. You also have an apotome on this system. So it takes a number of images with the grid projected onto the focal plane, 3 to 5 images. And from there, it uses a combination of these optics and mathematics to generate an optical section. And then you can use deconvolution to improve the resolution of your image a little bit further. So conventional, sectioned, and deconvolution will give you a slight increase in resolution to look at smaller structures in more detail. Mounting frames, you've got a number of different mounting frames that will go into the system. This is just an example of what you don't have all of these at the moment. You're still waiting on some to come in from ZEISS Germany. You'll have frames that just basically go into this mounting frame that's already on the system. So if you're not using that heated insert, you'll be using this one for plates and dishes, and these little inserts just go into that area. So it's just what's called a push and click system. So you easily should be able to exchange these in and out of that mounting frame, depending on what you want to look at. These are some optional accessories I just wanted to talk about. You don't have these, but these are things that you can get and maybe considered sometime in the future. If you want very, very precise temperature control, you can obtain a sensor which will actually directly go into the dish you're using itself. Okay, so typically speaking, you might run an experiment where you want the temperature really, really optimized. You can have a temperature sensor that goes either in the liquid or into the chamber area to measure the temperature in real time. If your samples are evaporating, I don't know whether any of you have used these before, but Colt foil from Pecon, it's basically a little foil cover that goes over your dishes. It's optically clear, so you can image in bright field and fluorescence. It's not suitable for DIC. It basically lets gases through, but water doesn't escape. So if you have evaporation issues at all and you can't resolve those, you can use Colt foil on your dishes to assist with that. I have used these ones and they're really excellent. These are little covers, little metal rings with a glass cover on the top. You can get those for both multi-well plates and for Petri 35mm and 60mm Petri dishes. You can autoclave them, but essentially, if you want to image DIC and make use of the system's capacity for rapid DIC bright field imaging and combine that with fluorescence, then you may need to think about these type of lids. Okay, they're not super expensive, but they do also help with minimizing evaporation as well. They've got like quite a nice seal on them. Do you have these available, these kind of glass covers, or do you normally just take the cover off? Yeah, so you can take the cover off, but for people doing long-term time-lapse imaging, you're going to get evaporation if you take the cover off. Okay, so these can assist with minimizing evaporation, and they're great for DIC. Perfusion, if you look at the heated insert here, you've got these tube ability to put tubes in there. So you can get something like a POC mini cell cultivation system, they're called. They fit directly into the into that holder. So if you want to do perfusion experiments, they can be done using a peristaltic pump, using these particular perfusion inserts. Now you don't have these, I'm just telling you that the system's capable of doing that if you are looking at something like that in the future. Okay. Trigger control, as I mentioned ad nauseum before, by controlling all of these components for rapid image acquisition. We can also use that triggering box that you have available for, it's basically just an input-output box. You can use the software and that box for controlling external devices like pumps or pico spritzers and other things. Of course, if I tell you you can have perfusion experiments on the system, they'll probably freak out because you can spill media and everything into the system. So be careful of that, but I'm just saying that the software can interact or interact with third-party components like those using TTL trigger pulses. I just want to talk about the software. We're probably running out of time a little bit. So key applications on this particular system would include 2D multi-channel imaging, of course, DIC imaging and fluorescence, and combining those. Very fast 2D and 3D multi-channel time-lapse imaging. The system is able to go very fast, so make use of that. Multi-position experiments in 2D, 3D, and combining that with time-lapse. Very large tile scanning in 2D, 3D, bright field and fluorescence. 3D reconstructions like deconvolution and using the apotome. Advanced software acquisition and processing tools are also available. Customizable macros are available, and image analysis. Okay. So just quickly on the time series type of thing, you've got to choose the right objective. Try and optimize your light intensity. You can do that here in the software. You've probably seen that already, the ability to change the light intensity of the colibri per channel. Use triggering for fast acquisition in the software. You'll find that under the acquisition mode here. So you can be interactive or triggered, fast, or just normal. That's where you would find it in the software. Get to know your camera. So have a look at your the resolution, look at your frame size, binning, gain controls, and any other advanced parameters the camera might have, usually under model specific, under the acquisition mode here. So get to know your different camera parameters and how it behaves. In time series, you can start and stop at a given time of day if you wanted. Experiment with different camera settings here as well. So if you're doing single channel imaging and you want to go really, really fast, then you can use things like burst mode, which basically puts all of the camera data straight into RAM. Okay, so you can't really do a time series with it. It's just basically go and it will do the camera's maximal frame rate all into RAM, not onto the disk. So experiment with that as well. But just get to know how your camera works within time lapse and see if you can get the most out of the system that way. Okay. And of course, optimize your environmental parameters. Tiles and positions, the module, you probably, a lot of you will be familiar with this already. In live cell work, we can easily calibrate our templates, whether they're slides or dishes, okay? To be able to easily navigate around and set up multi-channel, multi-position or multi-tile experiments are easily done within the software, either on multi-well plates or slides. The ability to do preview scans and all of that would probably, I think, make the workflow quite easy. Some people might disagree, but it's a very powerful tool within the software for this type of live cell work. Also, on the bright field slide scanning side, this is a bit of a dual purpose system. You can put your regular H&Es on this system and do whole slide scanning experiments using the 705 color camera and the tiles and positions module. You can use autofocus or definite focus for the focusing very rapidly, and then stitch your images on the system itself, or also in the free software ZenLight. Experiment Designer is another software module that you have available to you. So if you're doing complicated experiments that aren't just the one type of experiment, like normally you would set up an experiment, it might be a 3-channel experiment and you just hit go and that's it. If you want to combine experiments together that are a little more complicated, you can do that using Experiment Designer. So it's basically just like a series of blocks. So you might have in the first block, you want to do a single channel, and the next part on the same sample, you want to do at a different time point, you want to do 3 channels, and then again, maybe at a different time point, a single channel, again, 3 channels, maybe last you might want to do a Z stack. It's basically the ability to put lots of different experiments together in one string, okay? So it's for doing non-homogeneous experiments, so heterogeneous experiments that are more complicated. Sometimes you might want to do like, say 2 channels, and at a certain time point, you might want to do a Z stack, okay, at a given time point. And that's where you can use Experiment Designer. It's again for complex applications. Apotome 2 I've already explained, but at the IMB for deconvolution, Okay, so for apotome, I would use for mostly fixed tissue work. Okay, for slide work, maybe not so much for live cell work, but deconvolution, you can certainly, if you've got your camera parameters correct, you can use deconvolution for your live cell imaging work for generating optical sections. At the IMB, you have microvolution deconvolution software, which runs on your server and makes use of all of the power that you have on that supercomputer. The ZEISS CZI file can be imported using bioformats. I don't think it's direct, but it's pretty close to being direct. This would be suitable for live cell and large data sets. But as I said, be aware of your camera settings and objective choice for optimal results. We do have image analysis on the system as well. So the way it sort of works in Zen, you have an image, you pre-process it, you do segmentation, which is the part that is probably most difficult. You can do that with traditional thresholding or also using machine learning. And then from there, you can obtain your features, define what you want to measure, and then collect your data. The segmentation part is probably the most difficult in respects to this. Moving on from that, one exciting module that you do have on the system is called guided acquisition. And this is actually the real-time use of image analysis whilst you're actually acquiring. So what does that actually mean for you? So you can basically take an overview scan of maybe a well or maybe multiple wells using a tile scan at a given magnification. You can then use image analysis in real time, or sort of real time, I guess. It'll say you're looking at, you've got a million cells and you're looking for 5 red cells in there and you don't want to actually hunt around manually. You're going to use image analysis to find those red cells automatically and then tell the microscope to do something about that. So it'll analyze that overview scan, detect the objects of interest automatically that you define. Okay, in this case, it's dividing mitotic cells. It'll feed back that data to the microscope, and then you can tell the microscope to complete another experiment that might be go around and find all of those red cells and do a Z stack at high magnification. Okay, and it will do all of that automatically. So you have that available to you as well. Okay, so it just involves working out the image analysis protocols for defining what you want to find, and then let the microscope find those objects of interest and obtain whatever you want automatically. Might be a Z stack, it might be high magnification, whatever. Okay, does that make sense? Basically, instead of you having to find it yourself, the microscope is going to do it for you using image analysis. So the software we've tried to create an open environment within Zen. So we like Zen to have the ability to be able to interact with third-party softwares. So the ability to interact with programs like MATLAB, Fiji, ImageJ, Amiro, for example. We have a particular macro environment that enables users to customize their workflows using Python scripting. It's a bit more of an advanced kind of feature in the software, but it's something that the guys at the IMB would like to sort of experiment with a little bit, and that's why it has the module on there. So if we look at this scripting, this what's called open application development, it uses basically Python scripts, iron Python scripts, which allow you to simplify or customize and automate your specific microscopy workflows, basically from acquisition to data processing as well. Okay, you can analyze and exchange data using this macro environment with other applications. So you can interact directly with Fiji and Cell Profiler or MATLAB, for example, and Excel. You can create smart experiments within your experimental protocol using something called experiment feedback, which will, similar to guided acquisition, it enables you to use online image analysis to modulate your acquisition on the fly. And an example of that would be if you have an object in the center of the field of view and it moves, you want the stage then to move back to the center. Okay, so it can track an object, for example, in real time on the microscope. It's generally for advanced users, I would say. We also offer customized solutions and support for this, mostly out of Germany and Singapore. You can find more information from how this all sort of works, basically on GitHub, and also there's some forums available on the internet as well to give you more information on how this works. Just to outline it in a little bit more detail, so the macro interface within the software uses Iron Python scripting. It has a recorder, an editor, and a debugger. So that recorder means that you can say you do something with the software, and if you have record, it will record those programmed commands that you could then build a script for and then apply later on automatically. You can actually import scripting from third-party solutions as well, and you can exchange data with other software packages directly whilst you're acquiring or processing images within Zen. Not everything can be recorded in the macro environment, I guess, but if there's not something that if there's something that can't be recorded, it doesn't mean there's not a command for it. Okay, you can always find commands. Well, the first thing you might do is look in the help file for a given command, or you can go online to the forum and ask someone, and someone usually will be able to tell you what command will work for that particular thing you want to do. So what actually can it do for you? You can basically write your own scripts so that you can tell the microscope to do something that's completely customized. You can use these scripts for image processing and analysis, and a lot of scripts are already installed. Okay, so there's a whole bunch of scripts that are already there, and some macros that are already available on the top toolbar of the instrument for, say, stitching your images after you acquire and something like that. So within the software, you have a little tab here called automation, and that automation tab allows you to run macros before an experiment or after an experiment. Okay, and things that I could think could be useful might be something like, okay, I've done a whole bunch of tile scanning. After my experiment, run macro X, which will stitch all my images together. Okay, so basically the macro environment is basically a way that you can smooth out your workflow a little bit by customizing a script to do something specifically for you. Another thing might be move the stage position to a particular location, change the objective, focus the sample, and then start the experiment automatically. Okay. Or it could be something like execute this image analysis script after I've completed my experiment and export that data to Excel straight away. And as I said, there's more information that you can find on GitHub and also from the OAD developer community forum. Experiment feedback, sort of touched on that before a little bit, but basically this allows us to run a script before our experiment started, monitor the acquisition directly, and then do something after the experiment that might be display our results or a data log file. And some examples could be something like, I've already described the field of view thing, but acquire a maximum of, say, 500 cells in each well of a 96 well plate until the experiment stopped, and then graph my results, or something like adapt the exposure time during the experiment. So it could be like this example of what the script might look like. Again, this is for advanced users. And I've seen people do this. Basically, they can get like pretty much a real-time graph of what's going on in their experiment, okay, as it's acquiring. So that's experimental feedback. Just almost finished. You guys wanted me to touch on Appear. So Appear is it's a ZEISS initiative, basically for cloud-based computing. So there's a whole bunch of microscopy tools available to you on Appear. It's been up for about a year now. As I said, it's completely free to join. It's essentially a platform where users can just collaborate together, essentially. It has a whole bunch of image processing and analysis tools available that are free. It's pretty much suitable for new and advanced users if you want. There are public workflows or public scripting available to anybody, or you can actually write your own and share it with other users as well, if you want. It can also be private. There's a big push at the moment for machine learning on the platform. So there's some machine learning tools available for free on the platform as well. If you're a ZEISS microscope user, there is a special module that you can run in Zen. If you don't want to run your Appear module in the cloud, and you've got very large data sets, you can run your Appear modules locally on your local machine using the Appear module. So just register at appear.com and check it out, I would recommend. It basically just allows you to really create and customize a workflow for yourself. I'll just show you the overall kind of gist of it. Basically, you can pick a module, build a workflow, and run the workflow in the cloud, and then the results can be downloaded to you. If you're new or you don't have any coding skills, you can use those public workflows. If you do have coding skills, you can write your own. Now, I don't have coding skills, but some people do, so have a look and give it a go. This is kind of how the workflow looks. But it's just a modular structure. Each module is built. You can then, what the guy's doing there is just basically image conversion, uploading the file, detecting objects, and he's just joining those things together to create a workflow. So each module does something individual. It might be import my image, it might be image analysis, blah, blah, blah. You join those together visually to create a workflow, and your images can just be uploaded to the cloud and results will come out the other end, okay? And this is an example of how that's worked here. So we've done some images, so looking at cancer cells, the image has been uploaded to Appear. Segmentation has been done in the cloud using machine learning, using the Appear, I guess an Appear module for segmentation. The segmented nuclei has been analyzed, and then a report's been generated, and that's all been done in the cloud. Lastly, and I've almost finished, don't forget ZenLight. I don't know whether you're using ZenLight to analyze any of your images. I would recommend that you try it. There's a tutorial that I've put together that's on the desktop of the system there. So you can just download Zen just generally from our website, or you've probably got it on your server there somewhere. You can run that on your laptops or whatever. It's not just an image viewer. It's basically a stripped down version of the software. You can do all sorts of things with it. You can optimize your images, annotate, do simple measurements. If you've got multi-scene images, you can split those up, batch process. It's got a great 3D viewer. You can export your images in whatever format. You can do things like sharpening, background subtraction, and denoise. And you can create image panels for publications. I would definitely use it. And if you're not using it, download it and try it out. That's pretty, okay. It does only work on Windows. Yeah, and that might be so. Yeah, just to summarize, you've got a great system there. It's perfectly set up for high-speed live cell work. You've got all the incubation components that you require. You can do bright field slide scanning, optical sectioning using the Apotome or Decon. A great range of mounting frames for dishes and slides and multi-well plates, ZenBlue obviously. And just remember, think about resolution, speed and sensitivity, and ask what you need to see, and always optimize your system for your experiments to get the best results. Sorry, that was a long presentation, but thanks for your attention.
So hopefully everyone can appreciate that it's a pretty complicated system, but with the Zen software, it's talk to us if you have specific imaging questions or experiment setups. But yeah, it's very flexible as well in the option. So especially with these advanced modules that we've added on to allow for really specific custom experiments. So we've had a few users do this on the Nikon system, and so they can be bridged across. Talk to us, we can these custom experiments. Does anyone have any questions for Mark, the expert, while he's here?